First, label your centrifuge tubes as indicated in Table 1 of your lab handout under the label column, and arrange them in the rack in the same order as listed. Using a 1 milliliter bulb pipette, add 1 milliliter of milk, formula, lactose, glucose, and water to the corresponding centrifuge tubes. Be sure to use a new pipette for each solution to avoid cross-contamination. Now let's add the lactase enzyme to our experimental samples. Before adding the enzyme, give the solution a gentle shake. Then, using a fresh pipette, add 0.5 milliliters of lactase to the centrifuge tube labeled Milk plus L. Be careful not to touch the tip of the pipette to the sample. Close the lid firmly until you hear a snap, and then invert the tube to mix the solution thoroughly. Place it back in the rack and start a 7-minute timer. Repeat this process for all the tubes labeled Formula plus L, Lactose plus L, and Water plus L. Next, we'll prepare the control samples by adding deionized water. Using a new pipette, add 0.5 milliliters of DI water to the tube labeled Milk plus W. Close the lid and invert it just like before. Repeat these steps for Formula plus W, Lactose plus W, and Glucose plus W. Take a paper towel and label it with Milk plus W, Milk plus L, Formula plus W, Formula plus L, Lactose plus W, Lactose plus L, Glucose plus W, and Water plus W. Leave enough space under each label to place your test strip for incubation. After 7 minutes, dip a glucose test strip into the tube labeled Milk plus W for 1 to 2 seconds. Then wipe off any excess on the edge of the tube. Lay the strip below the corresponding label on your paper towel and set a timer for 30 seconds. Continue this process for the remaining tubes. Once 30 seconds have passed, compare the color of each test strip to the color chart on the glucose test strip bottle.